it's a weird thing to think about, but we all existed as one single individual cell shortly after the sperm fertilized the egg in our mother's womb, more or less. And it's also crazy to think about that one fertilized cell has enough information inside of it to multiply and develop into all the different cells of your body. And it continues to leave that legacy as you continue to develop and age. The cells that haven't differentiated into new different types of cells are called stem cells. And there's a lot of research going into how we can use stem cells to as a medicinal tool in a branch of medicine called regenerative medicine. And we've already learned a lot about cells and about tissues and about the human body by studying these stem cells. Cells are the basic unit of life that includes, of course, us as humans, and there are some fundamental features that each cell has. The first is they have a plasma membrane, or also called the cellular membrane, which we'll talk about what it's made of in a minute. There is some inner supportive structures called the cytoskeleton floating around, or not necessarily floating, but within the cell in a fluid matrix called the cytoplasm. And finally, there are organelles within these cells, including in almost every human cell a nucleus. At least in some point of the development of the cell, there is a nucleus. But some mature cells do not have a nucleus, like blood cells. Each of these cells have different functions which have to do with their shape, with their anatomy, with their structure. And this is a theme in biology, that structure determines function. Like the way that a cell is shaped, or the emphasis in different organelles, that's going to directly relate to what it's doing in the body, what it's doing within the tissues that it is a part of. For example, you have a red blood cell, which is small, and it does not have a nucleus. It does not have a nucleus, which allows it to, one of the theories is, carry more oxygen. And it's small, so it can fit through very small capillaries. All right, so those are two functions which directly relate to its, or two features which directly relate to its function. There are skeletal muscle cells, which are cylindrical and long. They have multiple nuclei on the outside. This directly relates to their ability to contract. They need to be long in order to do that. You have neurons, which are also have these receptive features on one end and some sensory output features on the other. So you have the dendrites receiving signal and the axon sending the signal. Sperm cells as well. This is the only cell in the body that has a flagella. So it has independent movement, which is necessary for it to partake or be a part of fertilization. So this is a figure from the book, shows these different types of cells and also the germ layers in which they derived from. So germ layers are, after a zygote then differentiates, it starts early on in the development to form these germ layers, the ectoderm, mesoderm, and endoderm, as it becomes a blastocyst. And then it starts to gastrulate or form these embryonic layers. Well, each of those layers then becomes different types of cells and different types of tissues, different anatomical parts of the body. And these are the ones which we know where they belong. So if it becomes part of the mesoderm, it can become cardiac, skeletal, kidney cells, red blood cells, so on and so forth. So the embryonic layer is also important in understanding its development. All right, so let's go back to some of the features of the cell, starting with the cell membrane. The general characteristics of this membrane, obviously, it's the layer which separates the outside from the inside. All right, we have a controlled atmosphere within. Outside, not as controlled. So there are a lot of things going on outside in which the cell will respond to. Similar to a whole organism. So it's kind of like the skin of the cell. This then plasma membrane controls or regulates what's coming in and what's going out as much as possible. And in allowing itself to do that, it's just going in and out. It doesn't want to be super thick. If it was super thick, then it would be hard to get in and out of. So it is very thin, 5 to 10 nanometers thick, which you actually need an electron microscope in order to see. It doesn't show up visually on our light microscopes that we use in lab. Now the structure then of this cell membrane follows what we call the fluid mosaic model. It's fluid because even though we draw it in these 2D images where things are just kind of stuck where they are, that's not actually what happens. In actuality, these proteins and different phospholipids and glycolipids, all these things that are within this lipid membrane are in actuality moving and developing, and maybe some parts are being removed and some added in depending on the development and age of the cell. It's a mosaic in that it has a lot of different parts that are kind of put together. So some of these include proteins. You have a channel protein listed here. You have proteins that go inside or outside or span the whole cell. You've got glycoproteins, glycolipids, all these other things. The main composition, or the main 5 things, are: first, you have protein complexes. About 50% of the cell are these proteins. Another 20% are these lipid rafts, which contain your phospholipids, sphingolipids, which are very similar to phospholipids, and picket fences. And picket fences are the ends, essentially, of the cytoskeleton. So parts of the internal structure which then need to attach to different parts of the plasma membrane. You also have glycolipids, which are about 5%, and another structure, another molecule very similar in structure as far as having a hydrophobic and hydrophilic end, is cholesterol. About 20% of the cell membrane contains cholesterol, and it is there for structural support. Essentially, it kind of breaks apart the phospholipid tails so they don't pack together too tightly, and that allows for permeability within the plasma membrane. A phospholipid, I just mentioned its structure before, has a phosphorus head, which is hydrophilic, and it forms hydrogen bonds with water. That's what's going to make it hydrophilic. And it has these lipid tails, which are hydrophobic, so those point away from the water. And it's double layered, so you have another layer of phospholipids on the other side, and the tails point towards each other and the heads point away. Within these proteins, you have integral plasma membrane proteins. These are embedded across the membrane, so they make it all the way across. Or you can also have peripheral plasma membrane proteins, which are just on one side, either inside or outside. The functions of these plasma membrane proteins, there is some overlap between integral and peripheral, but we'll start with the integral plasma membrane proteins. They can be for transport. We're transporting something inside and outside of the cell. They can be receptors. They can be enzymes, which are important for metabolic pathways. They can also allow for cell-to-cell contact, and there are 3 types of cell-to-cell contact, each of which has a different function. You have your tight junctions, which is kind of looks like Velcro between 2 cells, and this allows for the cells to be so tightly packed together that they are essentially waterproof, so it doesn't let anything in between the cell. You can also have gap junctions. Gap junctions are channels which allow things to pass from one cell to another. And so these are important for communication, for coordinating regulation between 2 cells. And then we have desmosomes, and desmosomes are kind of like tight junctions in that they're fastening 2 cells together, but they are more for structural support. So you may have a tissue that experiences a lot of traumatic force, such as the heart cells, which are constantly beating. They need extra support to keep those cells together, and so desmosomes or anchoring junctions would be for those. Integral plasma membrane proteins can also be for cell identity. So by looking at these different proteins, the immune system may then better be able to decide, all right, this is a liver cell, we will leave it alone, or this is an infected cell, we will try to remove it from the body. All right, peripheral membrane proteins also can be for transport. They can kind of taxi through the cell. They can act as receptors. They can act as enzymes. They can be important for cell identity. And again, kind of the biggest difference there is they have a little different. They can't quite be a channel because they can't pass all the way across, and so they have a different mode for moving things in and out. But otherwise, their functions are very similar. Can be very similar. All right. So then our internal part of the cell, which is providing structure and support, is the cytoskeleton. The cytoskeleton has 3 different types of structural components. Microtubules, which are large and hollow tubes. You have intermediate filaments and microfilaments. And these are not only for structural support, but they may also be for shuttling something from one side of the cell to the other. You can follow this this part of the cytoskeleton, kind of like a railroad track. They can also help a cell contract and move and change in shape. And so there are lots of other functions other than just keeping the cell stagnant. The cytoplasm is a viscous, mostly water material, but it also has everything within the cell, or holds everything within the cell in the dissolved substances there that is important for the cell. So it may also contain proteins, salts, ions, sugars. It definitely will have these organelles floating around in there, such as ribosomes and lysosomes and so on and so forth. And it has inclusions, right? These are specific to cell type and include like droplets in fat cells of lipids. Okay. Organelles. Okay, we mentioned this is one of the important features of cells. It literally means small organs. These are the basic organelles which you've learned over and over again. You have the endoplasmic reticulum, which is important for transport, synthesis, and storage. If it's rough, it has ribosomes on it. If it's smooth, it does not. Essentially, you can make proteins and then make little vesicles for them to move throughout the cell. The Golgi apparatus does something similar. As it goes through the Golgi apparatus, the protein may be modified or structured differently and then sent off somewhere else. The Golgi apparatus can also make vesicles for other things other than proteins as well. Lysosomes are important for digesting old or damaged or infected organelles or parts of the cell. Mitochondria is important for cellular respiration. Peroxisomes are important for lipid metabolism and detoxifying harmful chemicals such as hydrogen peroxide and other free radicals. So this is then your cell. This is what you've hopefully studied before. If not, that's the detail in which you will need to know this for an exam. All right, so cells have what's called a resting potential. A potential means we're talking about a difference between the inside and outside of the cell. This resting potential, when we're talking about different chemicals and different ions have charges. So we're talking about a difference in charge between the inside and outside of the cell. And generally, the inside of the cell has more negative than the outside of the cell. And this is at about negative 70 millivolts. That changes depending on different types of cells, but most cells are at about negative 70. And this is assuming there are no active processes, so you aren't actively transporting something in or out of the cell, the channels are closed, and you have these proteins that are generally mostly negative on the inside of the cell, which are driving the overall charge. All right, so what does this mean and how is this set up? Well, again, it has to do with the ions. We've got sodium, calcium, chlorine, potassium, and then PR stands for proteins. Okay. On the inside of the cell, you have a larger concentration of negatively charged proteins, okay, and potassium. And these proteins are too big to leave the cell. All these other ions are so small that they can actually go in and out, but the proteins are stuck in there. So you have this large concentration of negatively charged proteins. That's going to affect the attraction of these ions to that, or repel that these ions to that negative charge. And so what does that then look like? Well, again, electrochemical, we're talking about attractive forces between the charge, positive and negative, and then the just concentration. So potassium, for example, it has a larger concentration inside of the cell. So naturally, it's going to want to go outside of the cell. But because it's positively charged, it's also attracted to the negative charge of these proteins, which are stuck inside. So there's a point at which that attraction is balanced by its desire to go down its concentration gradient. And that then balance is the electrochemical equilibrium. Okay, potassium, on the other hand, has the or same ion. Potassium then is also attracted inside of the cell due to this negatively charged proteins. So one of the ways in which the electrochemical equilibrium is maintained or balanced is through what's called the sodium-potassium pump. Now this is an active process because it's actually moving these ions against their concentration gradient, so it requires ATP. And what it does is it takes sodium from inside the cell and it moves it outside the cell. That requires ATP. And then at the opposite end, once it's open on the other side, it takes potassium and moves it inside the cell. And that keeps the concentration of potassium high inside the cell and sodium outside the cell. Okay, so when we put this all together, this is how it works. Potassium naturally flows out of the cell with its concentration gradient, from greater to lower, through leaky channels. So channels that don't require ATP but are just open enough to where potassium can leak out. And that's going to create an electrochemical balance at about negative 90 millivolts. Millivolts are a measurement of charge. Sodium naturally flows into leaky channels, even though slightly less permeable, towards the negative charge, making it less negative. And the sodium-potassium pumps then counteract those leaky channels to make the equilibrium slightly less negative at negative 70 millivolts. Okay? So by using these pumps, because you're pumping 3 charges in for every 2 you pump out, you create a more positive environment. So that's how it goes from -90 to -70. All right, so we talked about some passive and active transport mechanisms in this electrochemical equilibrium, and we're briefly going to review these. So passive transport means no ATP is required, which means something is going from greater to lower concentration, and that's just naturally occurs. If there's a bunch of stuff in one area, eventually it's going to spread out. The 4 different types are diffusion. Okay, that's just something going from greater to lesser concentration. Osmosis, which is water moving. So if you have substances that can't move, but they're in different concentrations, and they're in a solution, and the water can move, the water will move to balance that out instead of the substances. So that's what osmosis is. And osmosis then creates or can create what's called a hydrostatic pressure, okay, where the water moving in or out then causes a force on or pressure on the cell membrane. And then the osmotic pressure then is the tendency of that water to move in or out of the cell based on those concentration gradients. Aquaporins are very important in this process because they provide the channel by which water can move through. So solutions can be isotonic, hypertonic, and hypotonic. That is referring then to how much of a substance is in 2 relative solutions, 2 solutions that are next to each other and are only separated by a semi-permeable membrane. The opposite to these terms is osmotic, okay? And this is referring to the water concentration. So if you're in a hypertonic solution, that hypertonic solution is also hypoosmotic. Hypertonic means it has more of the solute. Hypoosmotic means it has less of the water. And so just by being hypertonic, you are by default also hypoosmotic. And the same is true for hypotonic and hyperosmotic. Now, I tend to just use one or the other because it can be very confusing. Generally, I like isotonic, hypertonic, and hypotonic. But if you see these other terms, that's what it's referring to. It's referring to the water, and it's the opposite of the tonic term. So here are 3 blood cells in different solutions. And so if this red blood cell is shriveling, that means it's losing water. That means it is in a hypertonic solution. So this solution probably has more salts in it, and thus the water is flowing through the aquaporins into the hypertonic solution, and this red blood cell is shriveling. The red blood cell itself would then be hypotonic. If a red blood cell is maintaining its shape, then it's in an isotonic solution. If it's bursting, that means there's too much water coming in. That means the cell itself is hypertonic, and thus water is going through the aquaporins into the cell, and it's creating too much hydrostatic pressure, and then it bursts. All right, passive transport is, we're moving on to our third one, bulk flow or filtration. This is where water is forced through a membrane due to high pressure from another source. So the best example of this is in blood vessels, such as the Bowman's capsule in the kidneys, where water is forced out of these blood vessels because of pressure from the heart. Okay, it's just being forced out. Here, where the water is being forced out, it does not require ATP. It's because of some other force further upstream. Facilitated diffusion then is the fourth one, and this is a diffusion rate with the aid of some sort of transport protein. So here we have a transport protein which allows this green substance through. That's a facilitated diffusion as it passes down its concentration gradient. but it doesn't require ATP to do that. So aquaporins are an example of facilitated osmosis, but the same concept. All right, if things require ATP, then we have what's called active transport. There are different forms. You may be using a piece of the membrane. If that's occurring, then you have what's called bulk or vesicular transport, and there are different types of these. The first one's called endocytosis, where you're bringing something into the cell, okay? And so this is where you have some substance, it's now being entrapped into this membrane and brought into the cell through what's called a vesicle. If it's a molecule of some sort, then it's called phagocytosis, and this can be a way in which a cell gets energy or eats, Pinocytosis then is just engulfing water, and then exocytosis is removing substances out of the cell. So here we have a large particle being phagocytized by this cell. So the cell membrane is bringing it in here. Here's just water, so this is pinocytosis. Here is an endocytosis, which is mediated by this receptor. Okay, so this is a receptor-mediated endocytosis, one way in which this process would be triggered. All right, we have a couple other ones. Primary active transport is where a transport protein, which needs ATP in order to work, moves substances against the concentration gradient. We already talked about one of these, the sodium-potassium pump. You can also have secondary active transport, where two substances move at the same time through a channel protein against its concentration gradient. And if they're going in the same direction, it's called cotransport or symport. And if they're going in opposite, it's called countertransport or antiport. All right, and that's the cell. That's the cell membrane and the important organelles of the cells, which will come up again multiple times as we talk about the different way systems work.